So far we've covered complete dominance, and we're going to cover in this section some types of Punnett squares that are going to deal with traits that are not complete, and we're going to talk about multiple allele traits as well. So covering a lot of different things in this video. We're going to talk about incomplete. Incomplete is when you have a heterozygous that has a blending between two homozygous colors. So for example, if we have a white flower and a purple flower, and they're both homozygous, we end up with a pink flower, a blended color for the heterozygote. The codominance is co means at the same time. So we're going to talk about blood type because blood is a multiple allele trait. There's not just two forms of a gene, there are three. And there's a difference between type A, type B, and when you put them together, it's type AB. It's something altogether different. It's not a blending of the traits. So we're going to talk about that in this lecture. Now, your book uses a different notation than I've seen used in other textbooks and other examples. So I don't want it to throw you off with the extra numbers in here. But in this example, we're talking about curly hair and straight hair. Let's denote that over here. I think that's going to make a difference if we pick this up and come over here. This will be our homozygous, H1H1. This is our heterozygous, H1H2. And this is our homozygous, H2H2. That may help a little bit. So both parents have wavy hair, and the possibility of their offspring then, this one's H1H1, so this one will have straight hair. This one, H1H2, is wavy, so I'm going to W for wavy, and H2H2 is curly. So you see that we have, at the extremes, the homozygous ones, and the combined in the middle are the heterozygous ones. So we have somewhere between curly and straight, which is just wavy, and that's what you see in an example of incomplete dominance. An example of an incomplete dominant disorder is familial hypercholesterolemia, which is FH, and it parallels the number of LDL cholesterol receptor proteins in the membrane, the plasma membrane. So just to show you, like it was with the straight and the curly, we have a homozygous individual down here that's normal. We have a homozygous individual down here which has a very high number, and our heterozygous number is going to be in between those two. It's not a complete dominance or recessiveness, it's incomplete. Now normally when we do problems with incomplete dominance, we will use, where we used H's with the 1s and 2s a while ago, we normally would have done that with S's and C's. So S is straight hair, C is curly hair, S is straight hair, C is curly hair because the parents had wavy hair. Let's go ahead and turn this into a Punnett square to help with that explanation. So if we were to replace H1 and H2, we would use S for straight, C for curly. So we know that this one would be straight. We know that SC would be wavy because it's a little bit of both. There's two wavies, and then one that would be curly. So but the notation for incomplete dominance and codominance is a little different than complete dominance. We'll talk about that more in class. Let's take a look at multiple allele inheritance. This is controlled by multiple alleles. So instead of one form or the other, it is several forms. And the example that we have for this is our blood type. So our blood type is I is the gene. It's called the I gene. And A is the allele, A, B, or nothing there at all. So A and B are dominant over I, which is IO. A and B are also codominant. So if you are an individual that has type A blood, and there's an easier way to write this as well, we say that your genotype is either AO or AA. If you're type B as an individual, your type is BO or BB, and both of those are still type B blood. If you're AB, that's exactly what you are, is AB. And if you're type O, that means that you don't have the A or the B gene. So just to keep this in mind again, it's ABO blood types. A and B are dominant over I, which is O, type O. This is the I gene, it's type O. A and B are codominant, so we see them together, not blended, but we see them at the same time. The Rh factor is inherited separately from the ABO blood group. We're going to do a couple of examples of blood typing at the end of this video, but let's go ahead and do one real quick. Let's say that the dad is type A, B, so he has both an A and a B to give, so he can either give an A or he can give a B in his sperm. For the females, let's say that she is a type O. So if she's type O, she's OO. That's the only way to get type O. So what are the possibilities for their offspring? We have AO, BO, AO, or BO. So in this instance, the phenotypic ratio would be type A to type B, and it would be a 1-to-1 ratio. The possible genotypes would be either AO or BO in this case, and that's a 1-to-1 ratio. So that's just an example of how we would look at a Punnett square using blood type. All right, let's go ahead and look at polygenic inheritance. This occurs when a trait is governed by 2 or more genes or sets of alleles. Dominant alleles have a quantitative effect on the phenotype, and these effects are additive, like skin color. So if we have an individual, in this case, the genotype is AABB, capital A, capital A, capital B, capital B, the more dominant genes you have, the more intense the skin color. So in this case, this individual will be very dark. Here we have a homozygous dominant and a heterozygous, or if you look at it this way, the first one homozygous or heterozygous, the second one homozygous. So we have one small letter in the whole mix. So we're not going to have very dark skin, it's just going to be dark. What if both are heterozygous? You carry traits on both of them that are heterozygous, or if you notice the other possibilities, the number of dominant alleles determines the color. So if we have 2 dominant alleles, no matter what the arrangement, it's a medium color skin type. In this example, we have 1 dominant allele only, and that's light, and no dominant alleles would be very light. So this is called a polygenic trait. Let's do some practice problems. And it's important when you get ready to do these that you know what type of dominance we're dealing with. Is it codominance? Is it incomplete dominance? Is it complete dominance? What are we dealing with? So if a person with straight hair marries someone with wavy hair, can they have a child with curly hair? All right, so on the other slide, what we had was straight hair, and we had curly hair, and the intermediate then was 1 straight, 1 curly, which means that this one is wavy. All right, so let's go ahead and do the Punnett square. We have straight hair person that marries someone with wavy hair. So here's our genotype is SC. So let's see if it's possible for them to have a child that has curly hair. So this individual can only give an S. This individual can give an S or a C. So when we fill out the Punnett square, we notice that, get it filled in real quick, we notice that they have a 50% chance of having an individual with straight hair and 50% having offspring, 50% chance of having offspring with wavy hair, but there's no chance at all to have a child with curly hair because the straight hair is going to give that gene, and we don't have 2 curlies to put together to get the curly hair. So the answer, can they have a child with curly hair? The answer on this one is nope. All right, the next practice problem says a child with type O blood is born to a mother with type A blood. What is the genotype of the child, the mother, and what are the possible genotypes of the father? So let's take a look at what would happen in this case. So the child is, and I'll write down child so we don't get it mixed up. The child is O, and we know that there's A, there's B, and there's type O blood, and we also know that O is recessive. So if the child is type O, then they have an O from mom and an O from dad because it's a recessive trait. And it's born to a mother with type A blood. Well, you know that if we put mom, let's put mom here. Whoops. If mom is type A blood, in order to give her child an O, she has to be type AO, not AA. So this is what has to happen. So the child doesn't get an A, so I'm just going to put a mark in those 2 boxes. But we know the child is type O. We also know that the child gets an O from the dad. So in order for this to be a possibility, in this case, for the child to be type O, and just even one, we know that they have to have at least one copy. The dad has to have at least one copy of the O gene. So what could this other one be up here? We don't know that this is O, but what could the father be? So could the father be a type AB? Well, obviously not, because they have to at least give one O for the child to be OO. So he couldn't be that. Could he be type A? Sure, as long as he's AO. Could he be type B? Sure, as long as he's BO. But those are the only two, other than just type O, that he could be in order to have an offspring with this individual, with a child with type O blood. Let's look at another practice problem. From the following blood types, determine which baby belongs to which parents. All right, so let's see. If baby 1 is type O, the only possibility, let's pick up a different color so you can see it. The only possibility for this child is to be type OO, because those are recessive. However, if a baby is type B, that baby can be type BO, or they can be type BB. So, Mr. Doe has type A, Mrs. Doe has type A. Is it possible for them to have a baby with type O blood? Well, in order to have a baby with type O blood, they would each have to be heterozygous. So if this is the first scenario, is it possible? Is it possible if they both have 2 copies of the A allele? No, not at all. But you can see if they are heterozygous, they can end up with a child with type O blood. What about Mr. Jones? Mr. Jones is type AB, so we know what his genotype is. And Mrs. Jones is type A, so she's either type A, or this could be an O. In either case, can that individual have a child that's B, type B? Well, if they're, this situation, we have AA, AB, AA, AB. So if the mom is homozygous A, then no, she couldn't. However, it is possible, let's do the eraser, it is possible for the mom to have been type A, and instead of her genotype being AA, it's AO, and in that case, mom could give an O. Oh, this should be a B. Made a mistake on that one. Let's go back. So if dad gives a B, mom gives an O, it is possible for the baby to have a type B blood. The last practice problem for today is dealing with polygenic traits, and it's controlled by 3 pairs of alleles, A's, B's, and C's. What are the 2 extreme genotypes for this trait? So the extreme genotypes are going to include the dominant allele for each of the 3 pairs of alleles. So in this case, we would have an individual that has 2 copies of big A's alleles, 2 copies of the big B alleles, and 2 copies of the big C alleles. And these are going to allow us the extreme on one end. And that means that the extreme on the other end is going to happen when all of the alleles that show up are recessive. The last that we're going to take a look at are environmental influences. Environmental factors can influence the expression of gene traits. In the case of height, differences in nutrition are one of the factors that bring about a bell-shaped curve. And you just have a visual representation of a graph. We have 5 foot tall here, we have 6'5" down here. The girls are in white, the boys are in blue. This way you see that it depends. We have a nice bell-shaped curve where we have the median in the middle here, and we have that around 5'6", 5'7". And this just shows us that the distribution for this trait, which is height in this case, it could be determined not only by what the genes code for, but also the nutrition that those individuals have received can have an influence on that as well. Color in Himalayan rabbits, temperature can affect the phenotype. And Himalayan rabbits are a little strange because if you make them cold or a part of them cold, they turn a darker color. So here they put a cold pack on the back of this Himalayan rabbit, and in a little while we end up with a brown colored coat. So that's an example of how temperature can affect the phenotype of an organism as well. That concludes this chapter.